West Nile Virus by Lacey Bernard,
Caleb Borden,
and Katie Kallenbach.
West Nile virus is a member of the flavivirus genus. It has a positive sense single-stranded RNA genome, and it is enveloped with an icosahedral nucleocapsid. It was first isolated in 1937 in the West Nile district of Uganda. Since its discovery, there have been outbreaks in Egypt, India, France, and South Africa in the 1950s and the 1980s. It was introduced to the Americas in 1999, most likely on a transatlantic flight from Israel to New York, most likely in the form of an infected mosquito trapped in a suitcase. This is considered an involving host-virus interaction because the virus, through accidental relocation of a vector on an airplane, was introduced to a new geographic location. The natural reservoir for West Nile virus is birds, like crows and jays. After the bird is bitten by the mosquito, the virus replicates to high numbers and is then able to be transmitted through another mosquito to humans. The interactions between the virus and birds are an example of evolving host-virus interactions due to its instability and unpredictability. Infected birds are able to transmit the virus to other members of their population. Culex mosquitoes are the vector for the virus and take blood from infected birds to infect humans and horses. These are examples of dead-end hosts because only rarely can the virus be transmitted within the population. During the epidemic in the U.S., the virus was spread in blood transfusions, organ transplants, breast milk, and even one case of transplacental transmission until screening methods were developed in 2003. Prevention includes preventing any mosquito bites by wearing long sleeves and spraying with insect repellent, and reducing their populations by cleaning up any stagnant water and setting mosquito traps.
After a person is bitten by an infected mosquito, the virus replicates at the site of the bite and then drains to the lymph nodes. From here, it enters the bloodstream and eventually moves through the blood-brain barrier to infect neurons, causing neuroinvasive diseases such as meningitis, encephalitis, and paralysis. Neuroinvasive infections are rare, only occurring in less than 1% of those infected. However, those that are homozygous for the CCR5 mutation are at a greater risk of developing the neuroinvasive infection of West Nile virus. Most people are asymptomatic, and about 20% get flu-like symptoms. West Nile virus is able to evade the innate immune system by inhibiting type 1 interferon signaling. Currently, there are no treatments other than supportive care and no vaccines for humans, although there is a veterinary vaccine that has been developed and approved for use in horses.
We are seeing a national and global spread of West Nile virus. Globalization has contributed to the spread to new geographical regions, as we saw in West Nile virus's emergence in the United States. This image chronicles the spread over the first 5 years after West Nile virus arrived in the U.S. Within this time, all of the 48 mainland states had documented cases of symptomatic West Nile virus fever. The peak of confirmed cases was in 2003 with nearly 10,000 infected. This dropped down to around 2,000 after that, which is the average number of cases that we see most years until the present, with an outlier in 2012 with over 5,000 cases. The spread in the U.S. is mainly due to the effects of climate change. The increase in temperatures and more warm months has led to more infections because mosquitoes are not being killed off and their habitats are being expanded. The higher temperatures allow for the replication cycle to be completed more quickly. The expansion of warmer months also brings birds to their breeding grounds earlier. Precipitation also affects West Nile spread. Above average rainfall may lead to more water for breeding, but in dry or drought conditions, there is an increase in West Nile virus. The water sources are more likely to be stagnant, and the scarcity of water leads to a concentration of vector and host in the same place. We see an increase in the Dakotas, Texas, Louisiana, and Oklahoma as states that have the most infected people, which may be due to the bird migration routes that we see in the United States. The main points that we hope you take away from our presentation on West Nile virus is that the transmission of the virus involves multiple species. Our vector is a mosquito, with hosts being birds, which are the reservoir host, and humans and horses that are the dead-end hosts. Emergence and spread has been amplified by globalization and climate change. As West Nile virus fever spreads, in warmer, drier climates. There is no vaccine or antiviral treatment for the two forms of symptomatic West Nile virus. We see the neuroinvasive symptoms, which include encephalitis and meningitis in less than 1% of cases, and flu-like symptoms in roughly 20% of cases, which leaves nearly 80% of infections undiagnosed. And little is known to whether West Nile virus persists in the body after symptomatic periods or possible long-term effects.